Hello everyone, welcome back. So, in the previous lecture, we will discuss about drug discovery and development phases. So, before starting our today's topic, let's recap the previous lecture in short, in which we had discussed about the drug discovery. In that, we have talked about research and development processes, including the basic research as well as applied research. So, how the drug discovery happens, in that we came to know that various processes such as target identification, target validation, lead identification and lead optimization, and also discuss about the various departments that are involved in the drug discovery and development phase. So further moving ahead, let's talk about our today's topic, that is drug development processes. Once the lead optimization completed, the drug development process starts, which is further divided into two types, that is preclinical and clinical studies. So, former one is assessment of safety and efficacy in animals, while the latter is conducted in human volunteers and patients. So, let me talk about preclinical evaluation, in which we will discuss toxicological studies, pharmacodynamic study and pharmacokinetic study. So, the preclinical evaluation mainly consists of these three. So, first one is toxicological studies. These toxicological studies are performed to evaluate the toxicity of the lead molecule to a particular organ or to system or all over the body with respect to the duration and dose of the compound. So, it is conducted as acute, subacute, chronic or any other special toxicity, whether it is reproductive toxicity, mutagenicity or carcinogenicity. So, after toxicological study, we will further go for the pharmacodynamic study. It is the evaluation of the action produced by the lead molecule that is relevant to the proposed therapeutic use. The normal course of study is target to particular specialized condition followed by the organ-specific ex vivo or in vivo studies. Finally, the study is extended to the molecular level, like recording of receptor affinity, receptor binding studies on the cell membrane fraction from the organ to the cultured cells. Further, we will talk about pharmacokinetic studies. So, in pharmacokinetic study, the lead molecule emerged after above the two studies is subjected to the pharmacokinetic evaluation, in which we will discuss absorption, distribution, metabolism and elimination in different experimental animals. So, these are the three preclinical evaluation. After the successful completion of preclinical evaluation, we will go for the clinical trials. But before going for the clinical trials, we will talk about the various types of toxicity. So, various types of toxicities are acute toxicity, subacute toxicity, chronic toxicity, reproductive toxicity, carcinogenicity and mutagenicity. So, first one is acute toxicity, which is usually tested in two species by two routes and single dose to record the lethal dose in 50% of animals, and maximum tolerated dose can be determined in determination of acute toxicity. Further, we go for the subacute toxicity. Subacute toxicity is performed to identify the toxicity on longer use, that is for 1 to 3 months. Generally, 2 species and 3 doses are required to generalize the subacute toxicity. If we talk about the chronic toxicity is performed to identify the toxicity upon the long-term use, that is more than 6 months, usually tested in rodents as well as non-rodent species. Chronic toxicity study will perform simultaneously with the clinical trials. If we talk about reproductive toxicity is to record on reproductive system like reproduction, pregnancy or any other birth defects. If we talk about carcinogenicity, this toxicity study is to check the carcinogenic potential of the drug. It is conducted when drug is intended for long-term use, and for the usually 2 species are used and tested for at least 2 years. If we talk about mutagenicity, it is to evaluate the effect on genetic stability and mutation in bacteria and also in mammalian cells in the cell culture lab. To test the mutagenicity, usually in rodents and non-rodents are used. So once the preclinical data shows the promising results, the lead molecule is then tested in human volunteers and in patient, and it is called as clinical trials or human trials. So if we talk about clinical trials, as we all know that clinical trial consists of 4 phases, that is phase 1, 2, 3 and 4. So before proceeding for the clinical trials, the investigational new drug application is applied to the regulatory authorities, whether if we talk about the United States, it is Food and Drug Administration, whereas if we talk about the Indian regulation, so in India it is Drug Controller General, Government of India, that is called as DCGI. So the IND application consists of preclinical data, chemical structure, source, manufacturing details of the drug molecule, its specification of the dosage form and investigational protocol. So details of investigation involved inform consents of participants and any other agreements, etc. So the entire clinical trial is expected to achieve a well balance between the clinical, ethical and scientific issues. The ethical aspect look for the maintenance of right of autonomy of participant, that is healthy volunteers or might be patient, whereas scientific section is dedicated to assume the authenticity and statistical significance of the generated data. An institutional review board or ethical advisory committee or ethical advisory board, that are independent committees consisting of the physician, community advocates, socially aware personnel. These personnel are responsible to protect the right of study of participants. For any IND protocol, institutional review board approval in addition to the permission from regulatory authority is mandatory. So the clinical trial consists of 4 phases which is with a specific objective. If we talk about the phase 1 clinical trial, so it is conducted in small number of healthy volunteers except in specific conditions like testing of anti-cancer drug where significant toxicity is expected. So in case of anti-cancer drug, we will not go for the healthy human volunteers, that usually range from 20 to 100 participant for the short duration of action, short duration of compound administration. The entire study usually completed within 6 to 9 months. So 6 to 9 months is the duration for phase 1 clinical trial. So the major objective of this phase include the determination of pharmacokinetic difference between the animals and the humans, because we perform the preclinical evaluation in the animals. So here is the here we calculate the pharmacokinetic difference between the animals and humans and also determine the safe and tolerable dose to the humans and its documentation of any predictable toxicity. In phase 2 studies, these phases designed to determine the effectiveness as well as safety of the drug candidate in patient with the targeted disease. That means in this study we will use patients. So based upon the type of drug candidate, the condition it treat, the duration range between the 6 months and up to the 3 years depending on the which drug we will use. So if we talk about phase 2, so phase 2 is further divided into 2 phases, that is early phase and late phase. If we talk about early phase, it is conducted in comparatively small number of patients, that is ranging between 20 to 200, and mainly observe the potential benefit versus risk assessment. If we further talk about late phase of phase 2, it is conducted in comparatively large number of patients, that is ranging between 50 to 300, to ensure the safety as well as efficacy of the drug candidate in the patient. So the entire phase 2 may include the placebo, marketed standards as well as the drug or both groups for the comparison. So after successful completion of phase 2, which is a very long term of period. After that we will go for the phase 3 clinical trial. This is conducted in still more larger number of patients, that is ranging between 200 to 1000 plus, to further establish the safety and effectiveness of the drug candidate. The minimization of error on the data generated in phase 1 and 2, these phase 3 studies, another objective, the usual duration of this phase is between 1 to 4 years. So, upon completion of phase 3 studies, the sponsor files the NDA, that is new drug application, with respective regulatory authorities to market this drug. The NDA consists of complete monograph of the product, proposed brand name, package inserts, detailed drug development data, etc. These application is then further thoroughly reviewed by the regulatory department. There may be several correspondence between the sponsor and the regulatory department for the clarification when it is required. Once the NDA is acceptable, then the drug enters in the market with the new drug status. That means on the label it is clearly specified that is this drug is available for the controlled marketing. So this phase is further extended to phase 3B, which is usually begins for approval to supplement additional safety data. In some cases, it may be conducted upon approval of drug to produce the more data that may prove the usefulness. It also includes comparative analysis about the cost and effectiveness of the drug against the previously available marketed drugs. So, after completion of phase 3 and filing NDA application, the drug is approved for the phase 4 trials, that is post-marketing surveillance, and hence generally do but have specific time period. It is mainly focused on identification of rare side effects, previously known side effects, and even therapeutic indications. So these are the phase 1 to phase 4 clinical trials. So after completion of phase 1 to phase 4 clinical trial, let us discuss what are the objective of these phases. So in phase 1, we determine the pharmacokinetic profile of the drug to ensure the safety of the drug and predict the various toxicities. After completion of phase 2, in phase 2 we will talk about the safety and effectiveness of the drug molecule and also determine the short-term side effect of that particular compound, and also decide the doses regimen, or we can say dose range. So after phase 2, in phase 3 we will discuss risk versus benefit ratio or any other required labeling information, and also assess the less common side effect which is generally not noted down in phase 1 and phase 2. So after completion of the clinical trials, let's discuss about non-clinical development activities. It is simultaneously developed when clinical trials are conducted. So the innovator company also carries out number of non-clinical development activities while the clinical studies are performed. These research activities are essentially carried out in-house and includes studies like establishment of pharmacokinetic and pharmacodynamic, that is ADMA, in laboratory scale. Also assess the safety and efficacy whenever possible, and also develop the formulation for the clinical trials, develop the analytical and chemical methods for the evaluation, and also evaluate the product stability and develop the manufacturing processing. So the non-clinical research activities are of paramount importance as they support the clinical studies and commercialization of new drug, also depend upon the success of these studies. Thank you.